Welcome to our Anne Klibanski visiting lecture. I'm Miriam Bradella, the director of the Center for Faculty Development. The goal of this lecture is really to advance women faculty across the MGH, the US, and the world. And before we start with our main lecture, I would like to briefly say a few words about the origin of this lecture series. So the Anne Klibanski Visiting Scholar Award was established because as we all know, women are hit disproportionately by the COVID-19 pandemic with added childcare and household responsibilities. Women lag behind men in academic promotion and leadership positions. And one reason for the lower promotion rates of women are difficulties to travel for national and international presentations. And now with the COVID-19 related travel bans, most grand rounds in visiting professorships are being conducted virtually, which provides a great opportunity to give national and international talks from the comfort of your office or living room. So the Center for Faculty Development's Anne Klibanski Visiting Scholars Award organized visiting professorships at national or international institutions for 36 women faculty and postdocs at MGH who have shown exceptional promise as leaders in their field. And a couple of words about Anne Klibanski, after whom the award was named. Dr. Klibanski is the president and CEO of Mass General Brigham and the former director of the CFD, so my predecessor, and she has been a strong supporter and advocate for women physicians and scientists at MGH, including myself. And among many other initiatives, she established the MGH Backup Childcare Center and the Claflin Awards. And they support the careers of female scientists. And before Anne became the CEO, she was the chief academic officer at Mass General Brigham and also was the head of the neuroendocrine unit and an extremely accomplished NIH-funded researcher. This is just my mouse here is very sensitive, an overview of the Anne Klibanski Scholars. And here are the organization where they're giving lectures. And I just want to highlight the Erasmus University of Rotterdam because of our guest speaker today. So we are trying now with the Anne Klibanski Visiting Lecture, we are trying to bring together faculty from our institution and the host institutions that hosted our scholars to give a talk that brings together the expertises of both scholars. And we're trying to also create new connections and communities and collaborations with these lectures. And these lectures are open to both the MGH and Greater Boston community and that of the host institutions. I will introduce our speakers today, and then we will have, I think we're going to change the order. We're going to have Esther start, then Lilla, and then we will save the questions for the end. So today we have the pleasure to host Esther Bron from Erasmus University Medical Center from Rotterdam, the Netherlands, and Lilla Zöllei from the Department of Radiology here at MGH. And as I said, I'm going to do the introduction first, and then we will start with the lecture. Lilla Zöllei is assistant professor of radiology at Harvard Medical School and assistant in neuroscience at MGH, and she has a secondary academic appointment through newborn medicine at Boston Children's Hospital. Her scientific investigation focuses on pediatric MR imaging and developing computational tools that are capable of exploring the dynamic aspect of neonatal neurodevelopment. And she's the recipient of multiple NIH grants on this topic, and she obtained her PhD in computer science at MIT. Esther Bron is also assistant professor in the Department of Radiology and Nuclear Medicine at the Erasmus University Medical Center in Rotterdam in the Netherlands. And she's the PI of the research line Neuroimage Analysis and Machine Learning. Her mission is to ensure an accurate and early diagnosis and eventually the right treatment for the full spectrum of patients with neurological disease by optimally combining brain imaging, big data, and artificial intelligence. So without further ado, I turn it over to our speakers.
So yes.
Thank you.
Thank you for the introduction, Miriam. I'm a bit of improvising here because I'm now first. So Lilla was going to talk about neurodevelopment, and my part of the talk is mostly about neurodegeneration on brain imaging using machine learning. And I would like to tell you a bit more about my work and convince you that we need technology to improve early diagnosis of dementia And that's the way to do this. The key factor in making this happen is multidisciplinary collaboration. So first, a bit more about the background. So worldwide, about 50 million people are living with dementia. And the most common form of dementia is Alzheimer's disease, as you may know. But you may not know that it, on average, takes quite some time before people get a diagnosis. So in people with late onset dementia, so people that get dementia at an older age, it on average takes 2.8 years. And in people that get it at a younger age, it takes even a bit longer. And this, while we know from research, so there's a lot of research being done on this topic, obviously, we know that the disease already starts like 25 years before the onset of symptoms. And we can also see differences on MRI scans and in other measurements much earlier. So there is a huge window of opportunity to make this diagnosis earlier, to identify individuals at risk for developing dementia earlier. And why would we want to do this? So there are, in my opinion, two main reasons why we want to do early diagnosis and identify individuals at risk. And the first reason is for the patients and their caregivers themselves, because this is a period of a lot of uncertainty, and getting a diagnosis removes this uncertainty and would give them access to supportive therapies. And delays them going to a nursing home, and eventually would also save society costs. But the second reason is maybe even more important, or at least as important, because it is related to medication development. So currently there are no disease curing treatments, disease-changing treatments for dementia, for Alzheimer's disease. Well, a lot of clinical trials are being done. And one of the main reasons for that is thought to be like that individuals are included in clinical trials at a too late stage, when they have too advanced disease already and it cannot be changed anymore. So identifying individuals at risk early would help them to be included in clinical trials at the right time, and would also make clinical trials more efficiently in that way. So that's the two reasons why we want to do this. And I think we should use technology to do that. And the main technology that I'm talking about is machine learning. So from my field, I work in the Department of Radiology, and also in this. Yeah, so there's a lot of research being done on this, and brain imaging is the main biomarker that is being used, especially for machine learning. But it's often combined with other things. So measurements on daily lives of patients, information from cognitive tests, from which you can see two examples here, and information from lab tests. So like proteins that are measured in blood or in cerebrospinal fluid. And if these measurements are collected on many cases, many patients and healthy people, machine learning methods can be trained to classify, to distinguish disease from non-disease, and to identify individuals at risk. So this is a large research field. There are to date over 1,000 publications on this topic, and it really took off from 2008 onwards. So these are two publications from 2008, and it started exploding then because of technology, but also because larger databases of mainly MRI imaging data became available then. And you can see after that, many more followed. And also these are reporting quite high performances in the literature. So you would say that this works. However, it's not really used yet. So although technology is there, we don't see it used in clinical trials or clinical practice so much. And I think there are three main reasons why this is not happening. So a lot of development of this technology is done quite monodisciplinary. So based on imaging only, well, for clinical diagnosis, clinicians, neurologists take into account all kinds of information. And so that's the way to do it. And also, of course, the clinicians need to be involved. And for sure, they need to be involved to ask the right questions that we want AI to answer. because many of the publications, they answer like simple questions. Is the person healthy, or do they have Alzheimer's disease? But the situation is often more difficult than that. And also evaluation is often not done completely well. So data is often highly selected, while we need more evaluation on heterogeneous data. So this is a picture of the big hall of the hospital where I work, Erasmus MC in Rotterdam, the Netherlands. And since I work in a hospital, yes, most of you probably work in a hospital, so work with clinicians and who see their patients. And I find it really a pity that this technology is not finding its way to practice yet. So since, I find this important and I like solving puzzles, I so this puzzle is quite far already, but there are some important holes to fill. So one is the multidisciplinary part, the multi-domain biomarkers. We need more insight on how to integrate all this knowledge. And a second one is a broad population, so not a highly selected research population, but this method should work for everybody visiting a GP or a memory clinic. And that includes not only Alzheimer's disease, because you often don't know what the pathology is on beforehand, but also other types of dementia, such as vascular cognitive impairment should be taken into account. And validation, objective validation is also an important point. So papers can report high performances, but really what is the way, and will this generalize to a broader population is important. And open science can play a key role in that by sharing algorithms, sharing data to achieve this. So in this presentation, I would like to show 2 examples from my own research that I am trying to contribute to this. So this first example is focused only on MR imaging. So the most common approach in the literature is to use structural T1 weighted imaging, input it into a classifier. The most often one used is a support vector machine, and then to train it for dementia. So accuracies that are being reported, areas under the curve of about 90% for Alzheimer's disease versus healthy cognitively normal controls. And for mild cognitive impairment patients that convert to Alzheimer's disease, this is 75%. So quite high performances, but there are some open questions. So in the recent years, in the whole field of medical image analysis and computer science, there's really a trend towards using neural networks. So mainly convolutional neural networks. And for many applications, they clearly outperform conventional machine learning methods. And also in this field, people just started using neural networks, but I haven't seen any convincing work that shows that it really improves performance. So this was something I wanted to test. And I see a lot of, most papers are using the Alzheimer's Disease Neural Imaging Initiative data, which is really a great resource that really has brought this field forward. But not very often methods are evaluated outside this cohort. So I could find only 3 papers that did that, and the results varied a lot. So that body of work, so cross-cohorts validation of performance, is something I also wanted to address. So what I did is trained several machine learning pipelines on T1 weighted brain MRI data. And for convolutional neural networks, the assumption is that the machine learning algorithm can do the pre-processing, so that no pre-processing based on prior knowledge is needed. So that's what we tested. So one option, not sure if you can see my arrow, but here, minimal pre-processing, and the other option is pre-processing dedicated to dementia, computing a modulated gray matter map, which indicates the density of the gray matter in the brain. And both pre-processing methods we fed to support linear support vector machine classifier and all convolutional neural network classifier based on OPNI data to classify Alzheimer's disease patients and controls. So this is what the performance is. On the right here, you see the performance for the pre-processed images, and we see that pre-processing helps for both types of classifiers. And we see that the performance of both classifiers is exactly the same on this data. So maybe with more training data, CNN would outperform, but at least for this, for all the data we have available at the current moment, performance is the same. So we should not throw away the support vector machine as of yet. Also, if we look at the regions that are involved in the classification, so we made some maps for the support vector machine. I did that with support vector machine significance maps, which clearly show that the regions involved in making this decision are the hippocampus, medial temporal lobe, regions from which we know from prior knowledge from the literature that they are involved in dementia, in Alzheimer's disease mainly. And for the convolutional neural network, we made saliency maps, and there it's a bit less explainable. So they show regions in the cerebellum, in subcortical structures, and regions prone to white matter hyperintensities, but it's not so obvious as for the SVM, but still, of course, classification performance is the same. So then the second part of the question is the generalizability. So we used ODNI data for training, which is used a lot for machine learning studies. And in our institute, we have data available. So Erasmus CMC is also known for its population imaging studies, but we also have collaborations with the Alzheimer's Center and access in multiple multi-center projects to clinical data. And for this study, I used data from the Parelsnoer neurodegenerative diseases study, which includes, which is a collaboration between the 8 university medical centers in the Netherlands, data from their tertiary memory clinics. And it's the first time that this data has been used for such an analysis. So on the side, you see the results on the ODNI training data, and in the middle on the Parelsnoer data. And what really surprised me, I was happy to see that there was only a minor drop in performance when applying these classifiers trained on ODNI to our Dutch multicenter data set. So it dropped from an area, the curve of 94 to 90%, and while the data was quite different. So there were also similarities, but the largest difference is that for the ODNI, controls were really healthy. Well, for Parelsnoer, they were people that visited the memory clinic. They had subjective memory complaints, but still performance was quite high, so happy to see. And also for mild cognitive impairments, predicting who will convert to Alzheimer's disease, performance dropped a bit more, but also the data set was smaller, so which you can see reflected in the confidence intervals. So overall, a minor drop, which brings opportunities to apply these methods further and see their applicability to other applications. And I'm mainly interested at the moment to see if we can apply them in primary care as well. So in memory clinic, there is of course a lot of knowledge on how to diagnose dementia, but there's been some research that showed that also GPs and primary care physicians would like to have, as a lot of the diagnosis for dementia is being done in primary care, AI tools may help to strengthen their knowledge whether to refer patients or not. So that's something that I would like to look into in future projects. And next to support vector machines and deep learning networks, we could also look into more explainable machine learning methods, such as the discriminative event-based model that we developed that shows more the order in which events become abnormal. So this is a future direction. And for the second example that I want to show, that's about objective validation. So many methods, many papers are being published on this topic, and in biomedical image analysis, field, methods are usually compared at some point in grand challenges to have an objective comparison of methods. And in 2014, I therefore organized a grand challenge on dementia diagnosis, which was called the CAP Dementia Challenge. So we asked several research groups from different countries to train their methods on our multicenter, to apply their methods to our multicenter data set based on MRI imaging to classify Alzheimer's disease, mild cognitive impairments, and cognitively normal controls. And yeah, we were able to compare 29 methods from 15 international research groups. And so here you see an overview of the performances they achieved. And the main lesson that we learned was that the best methods combined several different measurements from MRI scans. So different aspects in the features was the most important aspect, and the classifier mattered less. And also we showed that this objective comparison was indeed important. And more recently, in collaboration with University College London, we organized another grand challenge to compare methods and the state of the art for Alzheimer's, and this was more about prediction. So are we able to identify how individuals will develop in 1 year from now? And this was based on all kinds of measurements. And in this challenge, so it was based on APNI data, but it was still a really objective validation, as follow-up data was collected after the deadline of the challenge. So nobody was able to cheat, if you want to put it that way. So an objective comparison, and we were able to compare 92 methods from 34 international teams. Methods were all really different. And participants were asked to predict diagnosis in 1 year, cognitive score, ADAS-Cog in 1 year, and an MRI measurement, the ventricle volume. And what we saw here is that methods could predict quite well diagnosis and the MRI measurement. However, prediction of the cognition was much more difficult. We made some simple methods, which are called benchmarks in this overview, and for the cognition measurements, none of the teams was really able to improve on that. And this is probably due to noise in this measurement and the short, so it's only 1 year follow-up time. So now we had in this project the 92 prediction methods, and of course their objective evaluation. But yeah, to use these algorithms further for further development, further validation, and see if they can apply it further, it is essential that these algorithms can be reused. So I set up a follow-on project to make these algorithms available. So that was a collaboration with the eScience Center to collect those algorithms and rewrite them in a standard format and make them easier for other users to apply. And as an added value to assess their generalizability on an external data set as well. And the main lesson learned, so this project ran in the last year and it's still running. We organized a hackathon, online hackathon last July with some of the developers of the algorithms. And the main lesson learned is sharing research code is really difficult. So especially this code that was developed for a challenge with a deadline, it's not the nicest code. It's quite complex algorithms. We really need the developers to be involved. So it was a much bigger effort than expected. And this validation study is still being continued. But for a next effort, a next challenge, because I think this is really valuable, I would like to take this into account from the beginning. So to also collect algorithms. So that brings me close to the end of my talk. So what I wanted to tell you is that that we need a more multidisciplinary approach to bring this early diagnosis technology, machine learning, to clinical practice and clinical trials. We need to ask the relevant methods, need to answer the relevant clinical questions, and objective validation, open science is very important. So yes, I want to thank the department. Thanks for giving me this opportunity to give this talk, and to the people from my group. And with that, I would like to thank you for listening.
Thank you so much, Esther. This was so impressive. Lilla, are you, yeah, you're ready, great. And we will, as I said, we will hold the questions for after the lecture. So we're gonna turn it over now to Lilla.
All right.
Perfect.
Thank you, Esther, for stepping in. And I'm going to jump to the other spectrum of brain development. I'm going to introduce you to some of the tools that we develop for the automated analysis of infant brain imaging data. I'm a computer scientist by training, so I'm interested in developing algorithms in order to efficiently, robustly, and accurately and quantitatively analyze medical images. And I've been focusing on pediatric brain MRI image analysis in the past 10, 15 years. So there's going to be three main algorithmic solutions or software tools that I would like to quickly introduce to you today. And at the end, I'm going to give you a glimpse into a couple of ongoing projects that are really exciting. And these are future projects that are ongoing currently in my lab. So the first tool or toolbox that I would like to introduce is called Infantry Surfer. The name comes from the fact that it focuses on infant brain image analysis. And FreeSurfer refers to the parent tool that has been developed in our lab for the past 20 years, I would say 20 plus years. It's freely available and open source, and it's widely used. So my goal was to develop a tool set, full brain segmentation and surface extraction tool set that's available for infants. I wanted to use the FreeSurfer format in order to be able to seamlessly incorporate outputs from my tools to future longitudinal analysis that is analyzable by the currently existing or the adult FreeSurfer pipeline. The current age range of interest is the 0 to 2 year age period, and the input images that we are currently analyzing are T1 weighted MRI scans. So this flowchart will give you a good idea of what the different analysis steps are in this pipeline. So we take our T1 weighted input image and we do some pre-processing to it. One key step is intensity normalization, and then we do skull stripping. Skull stripping is a process where we eliminate everything from the input image that does not belong to the cerebrum and cerebellum, and so that we can focus our analysis on the anatomy of interest. Then comes the volumetric segmentation step, where we assign an anatomical label to each voxel that belongs to that anatomy of interest. And then we do surface extraction. We are particularly interested in extracting representation for two surfaces. One is white and the other is PL surface. The white surface is the boundary that divides the white matter from the gray matter, and the PL surface separates the gray matter from CSF. And when these surfaces are created, or the surface representation is done, then we can also draw some labels, some cortical labels, and assign them to that representation. So the first key step is the skull stripping step. This is really important in order to be able to focus in on the anatomy of interest, as I mentioned. To this crowd, I probably don't need to emphasize how different the input images look like within our age range of interest. So within the first 2 years of life, the brain size can double in size. So there's size differences. There's intensity contrast differences on these images due to the ongoing myelination. And then there's also positional differences in case of infants, mostly the chin up and down movement, given that very small infants cannot be consistently placed in the scanner. So this is just to illustrate how resilient or how robust tools need to be in order to be able to analyze these infant brain images. Now, here, what I show you is 3 representative subjects from our cohort, a newborn, a 6-month-old, and an 18-month-old. And in the bottom row, we have corresponding manually labeled brain masks. So what we want to learn or automatically do is to get these brain masks when we have the input images. Our current solution is a deep learning solution that's included in our pipeline. So we have 3 fully trained convolutional neural nets with a unit architecture. These correspond to the 3 orthogonal views of these MR images. The outputs of these neural nets are linearly fused and thresholded, and that's how we arrive at our brain mass solution. Now, we skull stripped our data, and we are interested in we arrived at the skull stripping, and right now we are trying to do the full brain segmentation. The current approach that's implemented in our pipeline is a so-called Bayesian approach that again builds upon training data sets that were manually segmented, and we try to learn from this information. So our manually segmented data sets are spanning the age range of interest, so almost uniformly, as is indicated by this age chart here. And we chose a multi-level Atlas Fusion tool in our current implementation that allows us to combine training data with potential missing labels, and I'm going to show you why that's important, and also allows us not to assume any input intensity distribution about our input images. And again, I'm going to show you why that's important in the upcoming slides. So the 26 MR images that serve as our training data set are represented on this slide. Again, you can appreciate the huge variability in the intensity distribution in these images. And that's why it's important to have a tool that's robust to these changes or does not make any assumptions about them. This slide gives you the manually segmented versions of these images. And as you can see, in not all of the cases, we could separate out confidently gray and white matter areas. So that's why it was also important to have a tool that can accommodate missing labels in our training data set. The way our training, our segmentation works is the following. So we have our skull strip data in the bottom left of the image, and that we want to segment. So from our training data set, we, for computational reasons, we identify a subset of these images that are closest, and closest meaning either in age sense or in image property sense to this image. And spatially aligned, find the best spatial correspondence between these images and the new image. And when that's done, we bring all the manual labels corresponding to these training data sets into the space of this new test image. Now, using the intensity distribution and properties of our test image and the manually labeled training data set that's aligned to this space, we use some heuristics and come up with the final segmentation labels of our volume. On this slide, I show you some examples from the age range of interest with the manual segmentation next to the automated solution. So the middle rows are the manual segmentations. You see the full segmentation labels and the outlines, and then the automated solution. So when we evaluate our performance, we want to make sure that these two solutions are as closely matching to each other as possible. And the last final step of this pipeline is the surface extraction step. As I mentioned, we want to extract the white matter surface and the PL surfaces. If we have these surfaces, that allows us to compute key metrics such as the cortical thickness and gyrification patterns that have been shown to be key biomarkers at both of the age spectrums. And so these are important properties when we do group analysis, both when we are studying normal development and comparing healthy controls to disease groups. The top two rows show you a representation of the PL surface. In the middle row, I overlaid some cortical labels on these surfaces. While in the bottom row, we have the, so to speak, spherical representation of these surfaces. It's probably more intuitive for most of you to look at the first two rows, but mathematically speaking, or for mathematical point of view, we prefer the spherical representation. It allows us to do our computation in 2D instead of 3D, so makes it more efficient. But there is a one-to-one correspondence between these more intuitive and sort of mathematically more pleasing representations. So this pipeline is already available, and if you would like to test it out, it's freely available, it's open source. We try to be very responsive to our users, so we appreciate feature requests, feedback on on your experience. So if you are working in this field, please try it out and give us some feedback. The next tool or algorithmic solution I wanted to talk about is an atlas construction tool specifically designed for the neonatal population, and it's using an unsupervised learning framework. As I mentioned before, there's a rapid development of the cerebral cortex during the perinatal period. So static atlases cannot be used as a representation for these. And we need spatial temporal atlases that characterize this complex process. Unfortunately, there's still a limited availability of cortical surface atlases in the infant brain literature. Some studies or some attempts that have tried to cover this void fall into one of these three categories. They threshold the subpopulations and use kernel regression for creating the atlases, or they select a representative or a mean surface as an initial template, then run several iterative rounds of repeated registrations or spatial alignments to this initial template solution, and then update the definition of the current template. Or there are group registration methods that do find a global optimum solution, but their performance is limited by the definition of the subpopulation of the subjects. What we proposed in our recent work is a deep neural network solution that achieves end-to-end training for group registration, and at the same time produces temporal atlases given the group of input neonatal surfaces. This is the graphical representation of our atlas construction framework, which was built upon the voxelmorph framework of my colleague Adrian Dalka that was introduced in the volumetric domain. It has two key modules, an atlas inference module and the spheromorph module that takes care of the spherical registration or the spatial alignment, the spatial establishing spatial correspondences between spherical representations of input surfaces. So as you can see, the atlas inference module takes an attribute, an age attribute as an input, and outputs an atlas representation. And the spheromorph module takes in this atlas and the surfaces of an individual and outputs a warped atlas. We have written up the registration part of this framework and in a neuroimage paper. So if you're interested in details, you can read about it there, or I'm happy to answer questions at the end of this talk about that. When we construct a new atlas, convincing ourselves and also the users that this atlas space is the better atlas representation is quite difficult, and usually we do it in an indirect manner. I'll show you two examples how we do it. We use a segmentation task in order to show superiority of this new atlas construction, and also registration examples where we show that in this new atlas space, registration or spatial alignment can be done more accurately. For our experiments, we used two publicly available data sets. Luckily, in the infant literature, these are becoming more and more available. So one cohort is quite large. It's coming from the UK. It contains images of more than 500 infants that were imaged at the mean age of 39.9 weeks. And the other data set is much smaller. It comes from Melbourne. However, it does come with manual segmentations. So that allows us to do a lot of quantitative analysis of our performance. And as I said, in our experiments, we do two types of evaluation: spatial normalization and surface parcellation. I'm just going to show you some qualitative results so that you appreciate, what we can produce with this framework. So in the spatial normalization task, what we wanted to show is that in our new atlas space, we can register our surfaces more accurately than in currently available tools. So the figure shows you age-specific convexity atlases that were generated by our framework. And the inserts and the bottom row is a zoomed-in version of the features that were projected onto the spherical representation. Even though we didn't have longitudinal data in this in our input images, we can see longitudinal consistency in these atlases, which is very promising for our atlas construction methodology. In this second figure, I compare solutions that were provided by another publicly available tool from the UNC group. And so in the top rows, you see the UNC solution, and the bottom row is ours. So UNC ours, with features again overlaid on top of the spherical representation. In the first two rows, we have mean curvature, and in the bottom two rows, mean myelin values, which is, myelin is in quotations because it's basically the T2 weighted values over the T1 weighted values. Here, what I would like to, again, this is a qualitative representation of how well our atlas construction is working. In both of these examples, in case of both of the features and in all of the timestamps, if we look very closely, we get a more concise representation of these maps, meaning that we can achieve more precise and more accurate registration in these new atlas spaces. And the other indirect analysis that I wanted to show you is the parcellation accuracy. So what happens is that we use this new atlas space in order to guide further automated segmentation of these cortical areas. Here I show again the UNC atlas solution in one subject, our solution, and the manual solution. So again, just visually inspecting the data set, our solution is closer to the manual. And the reason why we really appreciate the Melbourne data set, because this can be actually quantified. So even quantitatively, we could show that our solution is closer to the manual one. Now, switching gears a little bit from purely T1 weighted or MPH data analysis, I'll introduce you to a tool that we call Traculina. So that's a diffusion weighted tractography tool. And Traculina stands for tracts constrained by underlying infant anatomy. It's an automated global probabilistic tractography tool that uses both anatomical and tractography priors. So the anatomical priors are coming from the volumetric segmentation that our Infantry Surfer pipeline can achieve. And the tractography priors come from manually segmented tractography solutions that I'm going to show you in just a second. This tool was particularly designed for newborns. And what it does is each time you see a new test image, it can reproduce a predetermined set of tracts in a probabilistic framework. So let me show you what that means. So again, Trackulina is a global tractography tool, which means that it always reconstructs an entire path between two regions. There's no concept of stopping midway or just producing half of a track. And as I mentioned previously, it uses anatomical priors. So here in the middle of the slide, you see some diffusion-weighted information, the anatomical information that comes from our segmentation, and the output is going to be this probabilistic representation of the track location. Trackulina does not allow for exploratory tractography studies. So if you have a tract that you are interested in studying that's not currently defined in our training data sets, this is not the tool to use. And I also just want to emphasize that, again, this is not a set of deterministic rules that, describes, which regions of interest track should be going through. The training dataset currently contains 12 major white matter pathways that are listed here. If collaborators are interested, this training dataset can definitely be augmented, and we used TrackWiz, again, a tool that's developed in our larger lab in order to do the manual segmentation for these tracts. This figure shows you a representative training subject that is used in Trackulina. So the tracts are color coded here. So a deterministic tractography solution was manually labeled and is going to be used as input to our tractography solution. When we are evaluating the automatically reconstructed results, basically, we want to get as close to possible to the manually identified track solutions. However, I want to point out that sometimes the correspondence is not going to be perfect, and that's not necessarily an error. So I'm showing you some examples here. On the top row, we have manually outlined solutions from deterministic tractography, and then at the bottom, we see the global tractography solutions. So the differences can result from the two different methodologies, deterministic and probabilistic. And as you can see, the probabilistic tractography, even if there's potentially noise or some artifacts in the data, it's going to reconstruct the probable location of the tract. And if there's really some noise or abnormality, then the scalar maps, the FA ADC maps that we can overlay on these areas will be indicative that there is subabnormality or difference between, for example, a normal cohort and the disease subject. And just two quick slides here, actually just one, because the movie is probably not going to work on this PDF version of my slides. I wanted to mention that we are very cognizant of the fact that we are very fortunate that we have access to very powerful computational resources, but we have collaborators who do not have that luxury. So we are working on making versions of our software solutions portable and cloud optimized so that, for example, our collaborators in South Africa can use these tools as well. What I wanted to show you, and unfortunately the movie is not going to be working, but I'm happy to send it to you if you're interested. Besides in vivo imaging, I became very interested in postmortem imaging samples. So the Martino Center has amazing resources, and we are able to acquire super high resolution structural and diffusion weighted images of infants' postmortem brains. And so we are interested, I'm interested in looking at analyzing those and studying healthy development and as well as SIDS. I wanted to emphasize, the set of collaborators who I work with on developing all these tools. And thank you again for your attention, and I'm happy to answer any questions.
Wow, thank you so much, Lilla and Esther. That was really amazing and so much data. And there have been several questions. So I will start because we are short on time. So Esther, I will start with you. Could you inform us which sequences are crucial in diagnosis and prognosis? In other words, could we reduce the number of sequences in patients from the memory clinic?
Yes, I can. Thanks, Ad, for the question. So structural T1 weighted imaging is absolutely crucial, I think. So most machine learning tools use that. But also FLAIR imaging is essential, especially as in my talk, we would like to go to a broader population, excluding and including vascular causes. FLAIR imaging is essential. So those two are absolutely essential. But in prior work, I've also shown that arterial spin labeling, so perfusion MRI and diffusion tensor imaging, for example, do have added value for personal diagnosis. Maybe not essential, but will improve accuracy.
Great. Another question for you. How early do you expect to see differences in the brain scan if a patient will go on to develop Alzheimer's? Do you need an early brain scan of the patient to determine changes with disease?
Yes, so there is evidence that it can be detected quite early. So there's a study from Diane, so dominantly inherited Alzheimer's disease that's shown group differences or changes over time, 20 to 25 years in MRI scans. And from our own group in the Rotterdam scan study, so Rotterdam study, population-based study where people are followed over time, we see that with a machine learning method, we can predict it 11 years before the disease onset. And that was on hippocampus only, so maybe whole brain it will even improve a bit. So between 15, 20 years in advance, it is detectable. Of course, closer to onset of symptoms, performances will go up, but yes.
Wow, thank you. So this is a question to both of you. This is a fabulous talk to both presenters. How is the concern of AI biases related to race, geography, gender, etc., are being addressed when training machines? That is such a relevant question, and it's, I think, in everyone's mind who's doing machine learning and AI.
I'm sorry.
Yes. Who's going first?
I can go very quickly. So, I mean, yes, we are very cognizant about that. And so when we are building our training data set, we try our best to include, large variability in the data cohorts, or if that initially didn't exist, test our tools or the differences in between outcomes when we do train on different types of data sets. So for us, it's really great to have the collaborators, for example, in South Africa, because we know that the cohort that's available there for the newborn subjects is very different than the local cohorts that we managed to recruit around Boston, for example. We are always trying to balance, female, the male population. But yes, I think it's a continued effort, and all of us need to be very cognizant to that and pay attention.
Yeah, I completely agree with that. I think we need more attention. And one of the points in my talk is that we need more validation studies and broader training data, because I definitely believe it's biased now. And we do indeed balance genders and age over controls and disease groups, but this balancing needs to be much broader than that. So there's definitely work to be done.
Thank you. Lilla, there is a very long question for you. Maybe I can read it, but yeah. Very exciting, but you might want to read it as well, because there is so much. Very exciting and important work. I have several questions related to FS. Is it working well in premature children, children over 2 years old, adults, and is it suitable, or do you plan to extend your work to this age range? 3, what about the performance in congenital abnormalities, corpus callosal agenesis? T1 weighted spin echo is changing in the first week. Will this have an effect on GM? wide matter.
Yes, so these are all excellent questions, and I'm going to try to answer very briefly, but I don't see the email address of the person who questioned, but I'm happy to, get in touch and email and kind of do sort of more detailed answer. Does it work well for premature children? We have tested it on some subset of it, but it is going to depend on how premature the children are and how and the quality of the input images. This is given that they are not well represented in the training data sets. We always. When I have collaborators asking this question, I always try to, ask for a couple sample data sets, and then we need to see, so it's by trial basis. Does it work with older, 2 years and older? Yes, we have tried it on, to up to 3 or 4 year olds. Now we are pushing down the applicability of the adult free surfer to 4.5 or 5 years of age. We are trying to push up, the age limit of the infant one. And so we are hoping to fill that gap. And I'm definitely working on collecting more training data sets in order to make the infants free surfer, span the 0 to 5 year age range. So to make that continuity even smoother. What about performance and congenital abnormalities? Again, we need to see how large these structural abnormalities are in the brains, and then there's definitely already intersubject variability in the normal cohort. And there's an intrinsic assumption that these are going to be representative of the new images that we see. So depending on how large these abnormalities are, the outcome is going to depend on that. And yes, the contrast definitely changes in the first week, but even in the first year quite significantly. So we do have issues, and we are constantly trying to improve our algorithms and atlases in order to get as good of a gray-white matter separation as possible.
Are motion artifacts an issue in these images? That's what.
Yes, I mean that's always the case, but especially in case of these kids. So it's actually newborns are actually more well behaved, or we have a higher success rate imaging them. It's easier at that age to feed them, swaddle them, and they fall asleep, and so they don't really move that much. When they get a little bit more active, so 6, 7, 8 months old, it's harder to do that trick with them. So we do see a lot of ringing artifacts, motion artifacts. So I have colleagues who are working on prospective, motion correction. There's a lot of research going into how to get better scans, but on our side, if there's a lot of motion artifact, then definitely the gray-white separation is going to be much more difficult or impossible to do accurately.
Thanks. Louisa, you have a question. You can just ask the question yourself.
Sure. Thank you. Yeah, thank you both for this talk. This is outstanding. And Esther, I commend you for your work in open science. I think that's just so great. And to get so many international groups working together is just outstanding. I just sort of, as a psychologist, I just wanted to follow up on your note about why it was difficult to predict cognition. You had said there was too much noise, even with a 1-year follow-up. Just wonder if you could speak a bit more to what the challenges were on that prediction challenge with cognition.
Yeah, thank you for the compliments first. Yes, so what basically turned out is that so we compared a lot of methods, and basically so there was one that performed better than just random guessing basically, but none of them performed well. Yeah, so I'm not an expert on these cognitive measurements, but we were expecting that we are planning to do, when ADNI data becomes available, a longer time follow-up, because we already collected longer term predictions. So we are thinking that over a longer time it might become more stable. But maybe, yeah, ADAS-Cog, maybe you know more about it than I do, just has a large variance over time in individuals.
It does. Yeah, I was hoping maybe we can talk offline more about how to make that prediction better. I'd be really curious to think about that, because we need that.
Yeah, I would be very interested.
One more question for you, Esther. Do you consider interpretability and explainability of these models as also as one of the missing pieces of the puzzle, or do you think it is not the main showstopper?
I think it's definitely important, but not the main showstopper. Yeah, so performance and showing performance in wide validation data, in representative data, I think that's the main key, and answering the right questions. And that is, I think, the key factor in building trust for clinicians. And of course, showing that this is indeed based on brain regions that we expect will further increase this knowledge and will increase the trust in the model. And of course, will help explaining to the patients. But I don't think we should wait for completely perfect explainable models. I think it can go in parallel.
Great, thank you. I think we are done. Thank you both so much. And Lilla, you handled this amazing, all this technical issues. True professional. Thank you, both of you. Thank you to our audience who from the Netherlands, now on and MGH. It's great to have you. And we have another joint lecture from Erasmus and MGH on March 30th. So I hope I see you all then. So thanks.
Our other Esther. Yes.
Thank you very much.
Thanks very much.
Bye. Thank you.